I'll be dealing with certain question from the chapter Molecular Basis of Inheritance. It's from class 12 NCERT. The first question is group the following nitrogenous bases and nucleosides. There are certain names given. We have to classify them as nitrogenous base and nucleosides. What are nitrogenous bases? The base which have nitrogen present in them. There are basically two types of nitrogenous bases, that are the purine and the pyrimidines. Purine is a 9-membered ring. It means there are 2 ring structures and total there are 9 members. Then similarly, pyrimidine is a 6-member ring. It has just 1 ring structure and has 6 carbon present in it. There are 2 types of purine, adenine and guanine, and pyrimidine are 3 types: cytosine, thymine and uracil. So the thymine is present only in case of DNA, and in case of RNA, thymine is replaced by uracil. Rest all the nitrogenous bases are similar in both DNA and RNA. So now that we know that there are 5 total type of nitrogenous bases, so we can easily can answer the question that among the names given, which of them are the nitrogenous bases. So the question's answer will be that adenine, cytosine, uracil and thymine are the nitrogenous bases. Now, which of them are nucleosides? What are nucleosides? It's basically composed of sugar and nitrogenous base. There are 2 types of sugar. One is the ribose sugar and the other is the deoxyribose sugar. Ribose sugar is present in RNA. Deoxyribose sugar is present in DNA. Ribose sugar and deoxyribose sugar differ at only one point, that the second carbon of ribose sugar has a hydroxy group present at it, whereas deoxyribose sugar does not have a hydroxy group on second carbon. Just that much is the difference between these ribose and deoxyribose. Otherwise, they are completely structurally similar. Now, that we know the definition of what is nucleoside exactly made up of. So what is we are going to deal with is that there are 2 names left, that is the cytidine and the other one is the guanosine. So cytidine is composed of ribose sugar and cytosine. Cytidine is present in RNA because it has ribose sugar, and along with them the nitrogenous base it has is cytosine. Now one more thing is that the nitrogenous base and the sugar are linked together by N-glycosidic linkage. There is a glycosidic bond between them. So in case of cytidine, the bond between the ribose sugar and cytosine is beta N1 glycosidic bond. So one nitrogenous the nucleoside, one nucleoside is clear. The other one is guanosine. Guanosine is composed of ribose sugar and guanine. Again, it is a part of RNA because it has ribose sugar in it. And the bond between the sugar and the guanine is beta N9 glycosidic linkage. Although this has nothing to do with the question of understanding that what are the linkages present, but just to make the things bit more clear. Otherwise, the answer is simple, that the adenine, thymine, uracil and the cytosine are the 4 nitrogenous bases, and cytidine and guanosine are the 2 nucleosides. The second question is, if a double-stranded DNA has 20% cytosine, calculate the percent of adenine in the DNA. We can answer this on basis of Chargaff's rule. Now, what is Chargaff's rule? It is the ratio between adenine and thymine and guanosine and cytosine, and it says that this ratio are constant and equal. To make it more simpler, what exactly this means is the amount of purine is equal to the amount of pyrimidine. That is, adenine and guanine, in the amount of adenine and guanine is same as the amount of cytosine and thymine or uracil in case of RNA. Now, what happens is that there is a base pairing rule also by Chargaff. It is a rule of base pairing is also given, which says that purine always pairs with pyrimidine. That is, there will be a bond between purine and pyrimidine. And to be specific, only adenine forms bond with thymine and guanine forms bond with cytosine. And in case of RNA, adenine forms bonds with uracil. So this is what Chargaff's rule says. So whenever there is a base this pairing or bonding between 2 bases, adenine pairs up with thymine and guanine pairs up with cytosine. So with these concepts, we can answer the question as cytosine is 20%, it's given in the question. So what will be the amount of guanine? It is 20% also. How? Because just now we dealt with that the purine bonds with the pyrimidine. So when cytosine is 20% and it has to bond with a purine, that is, it will bond with guanine because G and C forms bonding between them. So guanine also must be 20% so that all the cytosine gets to pair with the purine. So cytosine and guanine are equal in amount. So C and G forms bond. So when cytosine is 20%, guanine must be 20%. Then left out is 60% because 20% of cytosine, 20% of guanine is 40%. Rest is 60%. Now definitely the rest 60% will be either adenine and thymine in case of DNA and adenine and uracil in case of RNA. And now again adenine and thymine must be equal in amount so that a base pairing is formed between them at all points, and all the sequences adenine can pair up with thymine, they must be equal in amount. So for that, when 60% is left, it must be divided in 2 equal halves. So definitely adenine is going to be 30% and thymine is going to be rest 30%. And this way we can account for all the 100%. And the question which was asked to answer what is the amount of adenine, the answer is 30%. The third question is, if the sequence of one strand of DNA is given, we have a DNA sequence of one strand. What is the sequence on the complementary strand in 5' to 3' direction? What exactly is this 5' and 3' thing? There is a long stretch of DNA. In this long stretch of DNA, it has a 5' end and a 3' end. By 5' it means that the 5th carbon on one end has a free phosphate group, and by 3' it means that on the other end the 3rd carbon has a free hydroxy group. So that exactly is what the meaning of 5' and 3' that the 5th carbon on one hand has a phosphate group free, and on the other hand the 3rd carbon has a hydroxy group free. So when the sequence of 5' to 3' is known, we know that two strands having nucleotide sequence in such a way that they will form base pairing, because the two strands are complementary strands, and so they will have the sequences in such a way that base pairing can be formed, so that adenine can pair up with thymine and guanine can pair up with cytosine. So when the one strand has says that the first nucleotide base is nitrogenous base is adenine, so definitely on the complementary strand the base must be thymine, only then they can form a pair. Similarly, when the second one in the question, the second nitrogenous base is T, so on the complementary strand the nitrogenous base must be A, so that base pair can be formed. So just the complementary base will be present on the other strand. If the first strand has A, T, G, C, other must have T, A, C, G. Only then pairing can be formed between them. The two strands are complementary. So by this we can find out what should be the nucleotide sequence on the complementary strand, but the complementary strand is anti-parallel in direction. So in question, if we have the one strand which runs from 5' to 3', the other strand must be running from 3' to 5'. So now that doesn't answer the question, because question is asking that what will be the complementary strand sequence from 5' to 3'. That is very simple to be known, because once we have found out what is the sequence from 3' to 5' by the complementary strand rule, we can just turn around the structure. We have the base pairs, the base sequencing or nucleotide sequences from 3' to 5'. We just need to turn it around, and then we have the sequence of from 5' to 3'. That is given clearly in the slide. I have clearly mentioned that how it's been turning around and what is the sequence, the final sequence and the answer. The next question is, if the sequence of the coding strand in transcription unit is given, write down the sequence of mRNA. First, what is transcription unit? We know that a DNA transcribes to form RNA, but not the entire DNA can form RNA. there is a particular sequence on DNA which is known as gene that makes sense, and only that part of the DNA which is the gene which makes sense, only that particular part of DNA can then form RNA. Only that part of the DNA can then form RNA. So that gene will be, that part of the DNA which has gene in it will transcribe to form RNA. So it definitely, transcription unit must have structural gene. Other than that, what it should have a promoter and a terminator. That is not needed for this question. This question just you should know what is transcription unit. So it is basically a part of the DNA which has gene which can be transcribed to form RNA, and RNA then can form, by translation it can form protein. Now there are two strands of DNA. We know the one is from 3' to 5' and other is from 5' to 3'. The two strands, in case of transcription, the two strand, one will be called as template strand and the other will be called as the coding strand. The template strand is the one strand which will take part in transcription, whereas the other strand will not take part in transcription is the coding strand. Not both the, like in replication, both the strands of DNA take part, but in transcription both the strands of DNA does not take part. Only the strand of DNA which has polarity from 3' to 5' take parts in transcription, and the other strand which has polarity from 5' to 3' is known as the coding strand and does not take part in the transcription. So now one more thing is the 3' to 5', the strand which of DNA which has the sequence from the polarity from 3' to 5' on transcription, the RNA form will be complementary to the template strand. And also we know that the coding strand is also complementary to the template strand, because they both are two strands of DNA, both are complementary to each other. So the template strand is complementary to both RNA as well as to the coding strand, which means that this nucleotide sequence on the coding strand and mRNA will be same. In the question, we already have been given the nucleotide sequence of the coding strand. So that exactly is going to be, so what happens is that the coding strand sequence is known. So exactly that same will be the sequence for the mRNA. mRNA is a messenger RNA. On transcription, the RNA formed is, we have to basically find out what is the sequence on the RNA, which is same as the coding strand. Now the last question is, which property of DNA double helix led Watson and Crick to hypothesize the semi-conservative mode of DNA replication? We know that Watson and Crick have given the hypothesis of semi-conservative mode of DNA replication. This might seem a big term, that what exactly it is. I'll be telling you. First, we need to know that which was a property because of which Watson and Crick gave us the semi-conservative mode of DNA replication, and then I'll be telling what this semi-conservative theory means. The property was the property of DNA double helix, that they run in anti-parallel direction and that there is a bond between base pair of two strands such that adenine always pairs with thymine and guanine always pairs with cytosine, which makes them complementary. So basically this property, that both the DNA helix are anti-parallel, they have complementary base pair, they're complementary to each other because the base pairing is between them, adenine pairs with thymine, guanine pairs with cytosine. So that is a property which gave this semi-conservative replication theory. What happens is, for this, both the complementary strands first get separated. Mainly we know that when the double helical structure, they have to separate, denaturation has to take place. So both the strands first get separated. When the two strands get separated, on the two strands are getting separated, I have mentioned the slide. See that there is a two strands, DNA strands, both of them has got separated. Now what happens is, on separation, now both the strands act as template strand or the parent strand, which will help in the synthesis of a new DNA strand, basically what we call as a replication. So then what happens is, the new strand then synthesized is known as the daughter strand. There is a template strand, both the DNA strand got separated, we have two template strand, the template strand helps in formation of new strand which is known as the daughter strand. So in this process, what we notice is that half of the parent DNA was conserved in each new molecule of DNA. This is what is a semi-conservative model. Then the new strand which is formed is also complementary to the parent strand, so that old strand can help us to determine the nucleotide sequence of the new strand. So because the new strand form is also complementary to the old strand or the parent strand, so if we know the sequence or the nucleotide sequence of the new parent strand, we can know what is the sequence on the new strand thus formed. So this is the semi conservative model. Thank you.